worked at Feinstein Institute of Medical Research under Dr. Chen-Yan Li in Cushing Neural Monitoring Laboratory. He will attend Binghamton University and possibly West Point this fall.
Okay. Good evening, ladies and gentlemen. So our project is called a novel self-friendly microfluidic device for inducing localized hypoxia. I know it's intimidating. So let's get to it. Oxygen. Oxygen is a very important molecule. Does anyone know why? Okay, oxygen. Oxygen for cellular respiration to produce energy. And there's a condition called hypoxia, which just transfers for low oxygen level. So hypo, low, oxygen, hypoxia. So in vitro cell culture just means that cells are being grown outside the body in cell culture plates. It would be in vivo if it was inside. So some facts about oxygen levels. In the atmosphere right now, it's 21%. So when we breathe in oxygen, the air, it's 21% oxygen. And in cell culture media, it's like this. It's 21% oxygen also. And inside the cells, it's less than or equal to 6%. And here's a picture of like an incubator and this and all. So hypoxia and cancer. Hypoxia is a growing field of study in biology right now. So cancer is it's like an epidemic where it's the second leading cause of death in the United States. 1 in every 4 person dies from cancer every year. So how is hypoxia related to cancer? Well, the body sends this protein called HIF-1 alpha that we don't really know what it does, but all we know is that there's a lot of HIF-1 alpha present in various cancers. So there's a relationship between HIF-1 alpha and cancer. So why is it necessary to develop in vitro techniques that can target HIF-1 alpha? So we need to invest our time on investigating the signal and how it affects cells, and eventually we can figure out what causes cancer. Okay, hypoxia in the brain. So cerebral hypoxia is a condition where there's not oxygen in the brain, and it can be caused by ischemia, which there's not enough blood flow in the brain. And that can lead to cell damage, brain cell damage, and death. And that it's really bad because once a brain cells die, they never regenerate. So investigating the role of hypoxia may help save lives of people who need organ transplants. This is because hypoxia also relates to inflammation. So inflammation is a big problem in organ transplants because if you get a new kidney, you can have inflammation and that could cause death. And so investigating the role of hypoxia may help save lives of people who need organ transplants, those who suffer from cerebral hypoxia, cancer, and other diseases. So what's the problem? So how can we study hypoxia? Well, we need to make a condition where there's low oxygen, but we can't do that since the atmosphere is 21%. So there's 3 techniques that we can use. One is a hypoxia chamber, which is kind of like this picture. And that's just cell cultures and that use gas diffusion to create low oxygen. But the problem is it takes 3 hours to create 1 oxygen tension. And they cannot reach extreme hypoxic levels. And so we use microfluidics to overcome those limitations. So microfluidic devices, what you see here, it's basically a small chip that uses chemical reactants to decrease low oxygen, such as. And the benefit of microfluidics is that you can mimic the cell tissues because it uses small fluids. And the problem with microfluidics is that it requires a lot of interconnections, as you can see here. And most researchers stay away from them because they're very complicated. So what we want is a device that can reach hypoxic levels in minutes, no complicated gas connections, and it can have a lot of oxygen tensions. So we came up with the idea of using enzymes. So what we use is glucose oxidase and catalase. This is kind of like a 3D model of it. Here's glucose oxidase, the orange, and the yellow could be catalase. And it's being held together by a matrix, which is kind of sad. So the way it works is you have glucose and oxygen in the cell medium. And glucose oxidase turns out to gluconic acid and hydrogen peroxide. This hydrogen peroxide is then decomposed to produce water and less oxygen. So in short, this enzyme can decrease oxygen from the cell medium. So here's kind of like the picture. So this was the cell culture. You have oxygen in the culture and glucose. And what comes in is 2 oxygen, and what comes out is 1 oxygen. So we had to test enzymes, and we used a fiber optic sensor to measure oxygen tension, and we used various solutions and an MTS assay to determine the effects of the byproducts of the enzymes. Because if you look back, there's a buildup of gluconic acid during the reaction. So the things that we're going to test is the oxygen tension versus distance, the response time, and cytotoxicity. It just means how toxic is the byproduct. Okay, so what we learned from this graph, here's oxygen tension in this time. Over time, you see that we're increasing the distance and the oxygen level is increasing. So there's it's about a 0.2% oxygen increase per 100 micrometer. So the further away you are from the enzyme, the higher the oxygen level. The closer you are, the more lower the oxygen is. Okay. So we also figured out the enzyme is very fast. It works within minutes versus hours versus the hypoxic chamber. So here it took 2% for it took 5 minutes and 20 seconds to reach 2% oxygen tension. So it's very quick. Last test, we tested the cell viability. So here's a control. This didn't have the enzyme. It's a 4 unit per microliter. That's the concentration of the enzyme, and 10 unit per microliter. So what we learned is that the cells actually did grow and survive in this condition, while a very extreme level of the enzymes can cause cell death. So, yeah. So we basically learned that this concentration is the best concentration to producing hypoxic levels. So now we're going to use these enzymes and utilize microfluidics at the same time so that we can replicate cell tissues. So this is like a microfluidic that I made. It's made of PDMS, which is like a polymer. And we use plasma machines and all those stuff. Okay. So here's like the design of my microfluidic device. There's basically 2 chambers. One would be an hypoxic chamber, and one would have the enzymes. And there would be a normoxic chamber, which wouldn't have it. And the reason why we have those 2 chambers is that we could test if, like, let's say the HIF1 alpha, we could test if it actually does, if there's a correlation between hypoxia and the signal. But you could really study the signal with this kind of design. So the way it works is that it's being held by a mesh, and so the cell media would go in the inlet and go throughout the outlet. And this is where you grow cells, basically. Okay. So conclusions: we created a microfluidic device that can mimic in vivo conditions and can induce hypoxic levels very quickly. It's also very, I guess, safe for cells in that they're not toxic. And so for future studies, we want to implement this on a 3D model of the brain. Yeah. Okay.
Why do you want to do it on the brain, on a 3D model of the brain? Could you just explain a little bit more?
Okay, so remember where we're talking about cerebral hypoxia?
Yeah.
Well, the brain is very